Okay, so we will be reviewing for lecture exam 4 for AMP 1. The chapters that are going to be covered are 11, 12, and 13. So 11 is fundamentals of the nervous system and nervous tissue. And then 12 and 13 get into the different divisions. So 12 is going to be the central nervous system, and then 13 is the peripheral. Okay, so this is one of like the basic things to know about the nervous system. And specifically, this image may show up on your exam. So this is just showing you the difference between the sensory and motor output, and then also the kind of order in which it goes from seeing either the stimulus or something that intrigues your brain, which would be the integration. So here we can sense, we see that glass of water and we realize, oh, we're thirsty. This sensory input is information gathered by sensory receptors about internal and external changes. So the internal change could be that you are thirsty. The integration is the processing and interpretation of the sensory input. So your brain is going to decide whether or not to pick up that glass of water and drink it. The motor output is going to be the activation of effector organs, muscle, and glands, and then produces a function. So the motor output would be picking up that glass of water. This is something that is voluntary. We can't just think about picking up a glass of water. We actually have to do the act of doing it. So this would also be utilizing our skeletal muscle because it is a voluntary action.
The interneurons, like the same one?
Interneurons are going to be like more complex, whereas the integration is just like your brain making a decision. The interneurons are going to be like the actual action that's occurring, whereas the integration is just going to be processing. Oh, I'm thirsty. I should probably pick that up and drink it. Okay. Okay. So continuing on with chapter 11, we talked about the two different divisions of the nervous system. We have the central nervous system and the peripheral. The central nervous system is going to be your brain and spinal cord, and this is also what we call the integration and control center. This is kind of like the basic thing we think about when we think of our nervous system, just because we know that our brain is controlling everything. The peripheral nervous system are your nerves and ganglia outside of your CNS. So this is everything besides your brain and spinal cord. Within our peripheral nervous system, which I'll just be referring to as PNS, we have two subdivisions. We have sensory, which is afferent, and motor, which is efferent with an E. Whenever I'm saying it, I really make it a point to like say efferent instead of efferent, just because it can be kind of confusing, afferent and efferent. So just make sure you know the A and the E. Again, afferent with an A is sensory, efferent with an E is motor. And then we also have two subdivisions of our motor or efferent division. We have the somatic nervous system. This is voluntary, and this is conducting impulses from your central nervous system to the skeletal muscles. And then we also have our autonomic nervous system, and this is involuntary. This is conducting impulses from the CNS to your cardiac muscles, smooth muscles, and glands. So within the peripheral nervous system, you're going to want to know the two subdivisions of the motor, and then knowing somatic nervous system, autonomic nervous system, and then knowing which is voluntary versus involuntary. And then additionally, we have in the sensory or afferent division, we have somatic and visceral sensory fibers. The somatic sensory fibers convey impulses from your skin, skeletal muscles, and joints to the central nervous system. And then your visceral sensory fibers convey impulses from your visceral organs to your central nervous system. And then again, the somatic would be voluntary, whereas the visceral are involuntary. And then lastly, again, the motor or efferent transmits impulses from the central nervous system to those effector organs. Okay, moving forward, we start talking about nervous tissue. So we have two principal cell types. We have neuroglia and neurons. So neuroglia are the small cells that surround and wrap the delicate neurons, whereas the neurons or the nerve cells are excitable cells that transmit the electrical signals. And then we get into more specifically, these are divided by CNS in blue and then PNS in this purple color down here. So within our central nervous system, we have astrocytes, which are the most abundant. These cling to neurons, synaptic endings, and capillaries. They support and brace neurons, and they play a role in exchange between capillaries and neurons. So the way this could be asked on your exam is, which of the following neuroglia are the most abundant in the central nervous system? Astrocytes. We also have microglia or microglial cells. These are what we called defensive cells, and these migrate toward injured neurons. We have ependymal cells. These line the central cavities of the brain and spinal column and form the barrier between CSF-filled central nervous system cavities. Remember, CSF is that cerebrospinal fluid. And then we have oligodendrocytes. These are processes of oligodendrocytes wrap the central nervous system nerve fibers and then form the myelin sheaths. And so again, these four in blue right here are found within your central nervous system. So again, that's your brain and spinal cord. In purple, we have the neuroglia that are found within the PNS. The first one are satellite cells. These these are similar to astrocytes and surround the neural cell bodies where in your PNS. The actual purpose for the satellite cells are unknown, but we do have them again in our peripheral nervous system. And then we have Schwann cells. These are again in our PNS and they produce myelin. These are similar to oligodendrocytes and vital to regeneration of damaged peripheral nerve fibers. And then if you're able to see it, this is what those kind of look like. This may be more in your lab, but I wouldn't be surprised if this showed up on your lecture exam. These up here are your astrocytes. We can see those capillaries here. And then the neuron. We have our microglial cells or microglia. The ependymal cells, you can see these can have cilia. And then where these sit in correlation to the brain or spinal cord. So again, this is going to be your ependymal cells. And then we have those oligodendrocytes here, which we said produce myelin, and here are those myelin sheaths. Okay, and then we start getting a little more specific about the types of neurons or what's going on inside of the two divisions of the nervous system. So neurons, these are the structural unit of the nervous system. This is a single nerve cell. A neuron cell body or soma is the biosynthetic center of the neuron. This is where protein synthesis occurs. And then specifically, there's a difference between what is found. So a collection of neuron cell bodies in the CNS is called a nucleus, whereas a collection of neuron cell bodies in your peripheral nervous system is a ganglion. So you are going to want to know the difference between the two and be able to identify which term belongs to which division. So nucleus, central, ganglion, peripheral. We also have tracts and nerves. So tracts are going to be a bundle of axons in the central nervous system, and a nerve is going to be a bundle of axons in your peripheral nervous system. And then we get into more anatomy. So dendrites, these are the receptive region of the neuron. So this is what is taking in the messages. This is conveying messages toward your cell body as graded potentials, which these are going to be the short distance signals. And then we have axons. These generate the nerve impulses, transmitting them along the axolemma to your axon terminal. The molecules and organelles are moved along the axons by motor proteins and cytoskeletal elements. So we have anterograde and retrograde. Anterograde is going to be away from the cell body, and then retrograde is going to be toward. And then we have our myelin sheath. Again, this is composed of myelin, protects and electrically insulates the axon. And then the myelin sheath also work to increase the speed of your nerve impulse transmission. If a fiber is non-myelinated, the process is going to be a lot slower. So it's still going to conduct it, it's just going to be slower. And then we also have types of neurons. So the way that we classify them are functionally and structurally. So functionally, we have sensory, motor, and interneuron. Sensory, or again, these are afferent. The neurons transmit impulses away from the sensory receptors to the central nervous system, whereas the motor or efferent neurons carry impulses away from the CNS to the effector organs. And then interneuron is exactly what it sounds like. These are neurons located between the sensory and motor neurons. We can also classify them structurally. So we have multipolar, bipolar, and unipolar. Multipolar have many processes and they kind of just look like those finger extensions. Multipolar are going to be the most abundant. And then they're also a major neuron within your central nervous system. Bipolar, these are going to have two processes. These are located within your eye. And then unipolar have a single process. Some examples of these are sensory receptors, and these are mainly found in your peripheral nervous system. Okay, and then we start getting into action potentials, which I know a lot of students have trouble with. It's really hard to like break it down into a few sentences. So there is a lot going on the actual slide. We're going to break it down. So action potentials, this is the principal way neurons send signals over long distances. They do this by generating and transmitting action potentials. Only cells with excitable membranes can generate action potentials. So only cells with excitable membranes, these can be neuron and muscle cells, can generate those action potentials. The big thing to know about action potentials is that it is a all or nothing process. So make sure you know that. The action potential begins and ends at the resting membrane potential. Depolarization, which is occurring right here, if you can see it, it's going to be this number 2 here. So right here, let me zoom in. At this resting state, there's no ions moving through this channel here. Our second, let's say, step is depolarization. This is caused by an influx of sodium flowing into the cell. Again, and then so the depolarization is after the resting state, the depolarization is at approximately 30 millivolts, and it's caused by that influx. Depolarization ends when the channels inactivate, and then that repolarization and hyperpolarization are caused by an efflux. So we're going back here. Depolarization is caused by sodium flowing into the cell. Once it kind of hits that 30 millivolt threshold, we go into our repolarization state, which is caused by potassium flowing out of the cell. And when you are kind of reading or going over anything within your exam and you see this Na plus and this K plus, make sure like in your brain you're saying sodium, potassium, because on your exam they may have it written out as those specific words instead of the way it's seen here. Okay, so like we said, after the depolarization ends, it ends when the channels inactivate. Repolarization and hyperpolarization are caused by an efflux. If the threshold is reached, then an action potential is generated. If that threshold is not reached, the depolarization remains local. So again, we had our repolarization caused by the potassium flowing out of the cell. Hyperpolarization is below the threshold. This is caused by potassium continuing to leave the cell. And then we're back at 1, which is our resting state. So we went resting, depolarization, we reached the 30 millivolts. The potassium started flowing out of the cell. So we hit repolarization, hyperpolarization, it fell beneath the threshold. And then after hyperpolarization, we're back at that resting state for that process to continue again. So remember, this has to be a continuous process and it's all or nothing. So if that threshold is not reached, the action potential is not generated because depolarization will remain local.
Like both the potassium and sodium.
Yes. Mhm. Okay, and then something we talked about in class, specifically my class, was nerve impulse propagation. During this process, each action potential provides the depolarizing stimulus for triggering an action potential in the next membrane patch. These regions that have just generated those action potentials are refractory. And because they're refractory, the nerve impulse propagates in one direction only. Okay, and then action potentials are independent of stimulus strength. The strong stimuli cause action potentials to be generated more frequently, but not with greater amplitude. So it doesn't matter how strong your stimulus is. Even if you have a super strong stimuli, the action potential is going to be generated more frequently or more often, but it's not going to be stronger, or it's not going to have more power. It's just going to be more times. That's not the right word, but more often. And then during the absolute refractory period, a neuron cannot respond to another stimulus because it is already generating an action potential. During the relative refractory period, the neuron's threshold is elevated because repolarization is ongoing. So I would make sure you're familiar with this image here. On the exam, whether that's lecture or lab, I can see them giving you this image and having you identify what's occurring at each of the numbers or which of the stages. Or they'll ask you, depolarization is caused by what flowing into the cell, and that would be that sodium. Okay, so now we're going to get into chapter 12. A lot of chapter 12 is brain anatomy as well as function. So we're only going to go over certain parts of it today. We're not going to be covering like the specific anatomy of the brain, but more of the functions of the structures. Okay, so our adult brain is divided into the cerebral hemispheres, the diencephalon, brainstem, and cerebellum. Our brain has 4 ventricles, which is filled with that cerebrospinal fluid. Each of the cerebral hemispheres receives sensory impulses from and dispatches motor impulses to the opposite side of the body. So if you've ever heard your left side of the brain controls your right side of the body and vice versa, this is where that's coming from. And then we do have specific areas of our brain that are responsible for motory, sensory, and association. Our motor areas are the primary motor and premotor cortexes or cortices of the frontal lobe. Our frontal eye field and Broca's area is the more popular one that we learn about. Sensory areas, we have the primary somatosensory cortex, and then the somatosensory association cortex in the parietal lobe. Our visual areas, visual sensory areas are located in our occipital lobe. Our olfactory and auditory areas in our temporal lobe. And then gustatory, visceral, and vestibular areas in our insula. And then we have our association areas. The anterior association area is in our frontal lobe, and the posterior and limbic association areas span several different of our brain lobes. So like I said, we're not going to go over the specific anatomy, but more what each of those structures do and the function of them. Okay, so we have first our diencephalon. This includes our thalamus, hypothalamus, epithalamus, and it also encloses the third ventricle, which again has or houses our cerebrospinal fluid. It flows through our ventricles. So first we have our thalamus. This is our major relay station for sensory impulses, inputs from subcortical motor nuclei, and impulses traveling to the association cortices or cortexes from our lower centers. We have our hypothalamus. This is an important control center of our autonomic nervous system and the limbic system. The hypothalamus maintains water balance and regulates thirst, our eating behavior, GI activity, body temperature, an activity of the anterior pituitary gland. Lastly, we have our epithalamus. The epithalamus contains the pineal gland or gives rise to the pineal gland. Does anybody know what the pineal gland secretes? It secretes something that helps us sleep. The epithalamus, which houses the pineal gland, secretes melatonin. This helps to regulate your sleep-wake cycle, and I can see that question coming up on the exam. So our pineal gland is actually really tiny, so it's that small part of our brain that's secreting that melatonin to help us regulate our sleep. So we're going to keep moving our way through the brain. Next, we have our midbrain. This contains bundles of nerve fibers. It joins the lower brainstem and spinal cord with the higher parts of the brain. So our midbrain is here. It has our pons and our medulla oblongata, as well as our cerebellum. So the pons relay impulses to and from the medulla oblongata and the cerebrum. The medulla oblongata is the enlarged portion of the spinal cord, extending from the pons, which are here, to your foramen magnum, which is that big hole at the bottom of your skull. The medulla oblongata is also a cardiac, vasomotor, and respiratory center. Specifically for cardiac, the medulla oblongata transmits impulses to the heart via the peripheral nerves. The vasomotor center, some cells transmit impulses to the smooth muscles and walls of your blood vessels. And then respiratory center functions with centers in the pons to regulate rate, rhythm, and depth of breathing. I believe this is another part. Yeah, this is going to be part of your brainstem. And then we move on to our cerebellum, which are on the side here. The cerebellum are responsible for processing and interpreting impulses from the motor cortex and sensory pathways and coordinates motor activity so that smooth, well-timed movements occur. So there are two lateral hemispheres of the cerebellum, which just means if you cut your brain right down the middle, you would have one on the right and one on the left. If you see the cerebellar peduncles or peduncles, these are part of the cerebellum that communicate with other parts of the central nervous system. And then within the cerebellum, if we were, again, to cut the brain in half, they have what we call arbor vitae, which is white matter within the cerebellum. And when you look at it directly, they kind of look like tree branches. So that's why they're called the arbor vitae. Okay, and then this is when we start to talk about the meninges. So we did talk about this in class a little bit. The meninges are beneath the bone, so it's really important. This image in particular is important because it will show up on your lab exam, but specifically for lecture, you do need to know where the cerebrospinal fluid is. So this top layer would be your skin. And then we have bone is right here. So this would be your skull. And then our three meninges are going to be dura mater, arachnoid mater, and then pia mater. The way to remember this and the order that it sits in your brain, this is your brain right here. So brain, pad, brain, pia, arachnoid, dura. So the maters are there to protect your brain. And we have all of these layers that you have to go through before reaching the brain. And that's why TBIs or any injury to your head in general is really important, especially if someone hits their head and ends up bleeding from their head. It's really important to get that checked right away because it has to go through all of these layers, which means something is not right inside of you if you are bleeding from your brain or from your head, sorry. So starting again, let's start deeper. So again, we have our brain. We're going to start at the pia mater. This is the innermost membrane, and it's the thin membrane which contains many nerves and blood vessels to nourish the underlying cells of your brain and spinal cord. So this is kind of that last layer of defense, and it is super thin, the pia mater. Next, we have the arachnoid mater. This is the middle membrane. It's the thin net-like membrane. If you look at it closely, it kind of resembles, you can think of it like a spider web. So think arachnoid, like a spider. The arachnoid mater spreads over the brain and spinal cord, but does not dip into the grooves or depressions on the surface of the brain. And then there's a really important space between the arachnoid mater and the pia mater. This is the subarachnoid space. This is super important and will probably be asked about on the exam. The subarachnoid space contains your cerebrospinal fluid, and you need to know that it is between arachnoid mater and pia mater. Specifically, it's going to be, if we're starting at the brain again, it's going to be brain, pia mater, subarachnoid space, arachnoid mater. And then again, the arachnoid mater contains that cerebrospinal fluid. And then we move into the dura mater. This is the outermost membrane. It's also the toughest layer of our meninges. It contains many blood vessels and nerves. And the dura mater is directly, the dura mater is directly attached to your bones of your skull. So these are super important to know, not only for your lab exam, because this specific image will show up on your lab, but specifically your lecture. They will ask about the different maters and then which of these spaces contains that cerebrospinal fluid. Again, that's that subarachnoid space. This specific image is in your textbook. Okay, and then we have some different structures of our brain that allow for higher mental functions. So first we have our limbic system. These are different structures that encircle the brainstem. This is it's called the emotional visceral brain, and it plays a role in your memory. So some people who suffer a TBI or some sort of head injury, and they lose control or they don't really feel emotion anymore, they can have some sort of injury or defect to their limbic system. In most people, the left hemisphere of your brain controls language, the language implementation system, which includes Broca and Wernicke's area in the basal nuclei, analyzes incoming and produces outgoing language. The opposite hemisphere deals with the nonverbal emotional content of language. So again, this is when we talked about being right brain, left brain, things like that. So language control is in one side of your brain, the emotional on the other side. Memory is the storage and retrieval of information. It is essential for learning and is a part of consciousness. And then memory storage has two stages, short-term memory and long-term memory. Okay, and then we get into chapter 13, which is the peripheral nervous system. I know my class, we didn't get all the way through, so I don't think I have everything included. But if you don't have the notes, feel free to email me and I can send them to you for chapters 11, 12, and 13, but specifically 13. So again, the peripheral nervous system has different divisions. We have sensory and motor, and then within motor, we have somatic and then autonomic. And then within those two, we have the sympathetic and parasympathetic. So the peripheral nervous system, these are sensory receptors, include the receptors for the general senses, pain, touch, pressure, and temperature receptors in the skin, as well as receptors in skeletal muscles and tendons in the visceral organs. They sense organs contain sensory receptors and other cells that serve the special senses. We have touched on this a bit when we did the chapter on skin. So some of these words or definitions that you hear should be familiar. So the types of receptors we have are classified according to our stimulus. These are detected as mechanoreceptors. These respond to touch, pressure, vibration, and stretch. We have thermoreceptors. These are sensitive to changes in temperature. Photoreceptors, these respond to light energy. Chemoreceptors respond to chemicals, so smell, taste, or changes in blood chemistry. We have the nociceptors. These are sensitive to pain-causing stimuli. So for an example, extreme heat or cold. Exteroceptors respond to stimuli arising outside of the body. So these are touch, pressure, pain. If you grab something that's hot, most special sense organs. Interoceptors detect stimuli originating from inside the body. And then the proprioceptors detect changes in movement, equilibrium, or body position found in the musculoskeletal organs and structures. Okay, and then the general sensory receptors are classified as free or encapsulated nerve endings of sensory neurons. The free endings are mainly receptors for temperature and pain, with two exceptions. The epithelial tactile complexes detect light pressure, and hair follicle receptors respond to hair deflection. The encapsulated endings, which are mechanoreceptors, include tactile corpuscles, lamellar corpuscles, bulbous corpuscles, muscle spindles, tendon organs, and joint kinesthetic receptors. So again, some of these words sound familiar. That's because we did look at these when we did chapter 5. I believe it's chapter 5. Okay, and then also in our peripheral nervous system, we have our cranial nerves, which are important. And for both lecture and lab, you're going to need to know the function for each of these. So not only the function, but also them in order by Roman numerals and their name. So I'm going to zoom in a bit here. What I suggest you do is learn them all by name first, and then you can come up with a mnemonic, or there's many mnemonics online for you to remember them. So in order, the first cranial nerve is your olfactory nerve. This plays a part in smell. 2, we have our optic nerves. Optic meaning vision. You'll also realize that what we have on one side, we also have on the other. So please keep in mind, we have 12 pairs of cranial nerves, 24 total. Number 3 is going to be the oculomotor. This is responsible for eye movement, which is kind of in the name, oculomotor. Trochlear nerves, these are responsible for vertical eye movement. Number 5, this is going to be your trigeminal. The way that I always remember it is kind of weird, but trigeminal, May is the 5th month, and my brother was born in May, and he's a Gemini. So that's the way I remember that. I really don't know anything about the signs, but that's the only way I remember. Trigeminal is 5. Abducens is 6. This one is responsible as well for eye movement. 7 is going to be your facial nerve. So this is responsible for face movement and taste. Number 8, yes, is going to be your vestibulocochlear nerve. I know that one's a little hard, vestibulocochlear. This is responsible for hearing and balance. Number 9 is your glossopharyngeal nerve. This is responsible for throat sensation, taste, and swallowing. Our vagus nerve is responsible for movement, sensation, and also plays a role in your abdominal organs. Our accessory nerve is responsible for neck movement. And then hypoglossal tongue movement is going to be 12. So I know you're wondering, how am I supposed to remember all of these and the order? The mnemonic I've seen is on occasion, our trusty truck acts funny, very good vehicle, anyhow. So olfactory, optic, oculomotor, on occasion, our trusty truck, trochlear, trigeminal. The way you would remember these two, again, because they both start with TR, trigeminal, Gemini, May is the 5th month. Trusty truck, acts funny, abducens facial, very good, vestibulo-glossopharyngeal, vehicle, anyhow. Specifically, like I said, you're gonna have to remember the name and the Roman numeral. Will be the first one. I'm terrible at Roman numerals, so I would just have to write out like all of them as best as I could and then go back and check myself. You'll also have to know the function for each of these. So whether that's sensory, motor, or both, there's another mnemonic for that. So S is going to stand for sensory, M is going to stand for motor, B is going to stand for both. So olfactory is going to be sensory, optic sensory, so on and so forth. Some say marry money, but my brother says big brains matter more. There's 12 words here. They each correspond to the 12 cranial nerves. So again, if you need help with the mnemonics, there's some online that may stick with your brain better. I've seen Harry Potter mnemonics. I've seen some that aren't really appropriate. Those tend to stick better with some people. So go to Google and try to find one that fits for you. Same thing with the function, whether that's sensory, motor, or both. Okay, and then I do have a game. What I did, since we are recording these now, is I have them printed in 111. If you want to come by and pick one up, if not, feel free to screenshot and then play it on your own. If you don't have the answers, you can Google them. But since we are recording this, I'm going to go ahead and not do this on camera. But we do have them in 111. And we also have on the back of this worksheet that I have, and it has like the same quiz for the cranial nerves. And then, so these are the practice exams that I'm talking about. But if you would like one for your lab exam. It should be up by next week. Thank you.